In this video, we learn about RNA and how it's made. All right, so as we begin looking at RNA specifically, our desired result for this portion of the video and lesson is describing how RNA differs from DNA. And we'll begin with the role of RNA. But before we begin, I'm going to ask you to do something for me. Take out a piece of paper and a pencil or a pen or a crayon, I don't really care, something you can write with. And I want you to write down what you know or what you think you might know about RNA. And then we're going to see how close you're. So go ahead and pause the video here. Take a few minutes to jot down some things you think you know or do know about RNA, and then we'll dive in. All right, well, welcome back. Hopefully you took a moment to do the little exercise that I asked you to do, writing down what you know about DNA. Now, by the end of the lesson, you should be able to compare and contrast RNA and DNA. And then in the next lesson and the next portion of the video, you'll be able to explain the process of transcription, which is how RNA is made. Now, each nucleotide in the DNA is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. Now, you should already know that, but what you should also know is that this is also true for RNA. But there are a few important differences between RNA and DNA that we need to talk about. The first difference is that the sugar is really the distinction when it comes to the abbreviations of DNA and RNA. Okay, so in RNA, the sugar is actually a ribose instead of a deoxyribose. The second difference that we need to note is that RNA is generally just a single strand. It's not a double strand or a double helix like DNA. Okay, and we're going to talk about why that is as we go through our lesson here today. Now, the third difference is that for every T or thymine in the DNA code, RNA has a U, a uracil. So this means if you're forming a complementary code where there is an A in DNA, the RNA would pair it with a U or uracil. Okay, now it's these chemical differences that make it very easy for enzymes in the cell to tell the difference between DNA and RNA. Now, there are 3 main types of RNA, and most of them are involved in the process of synthesizing proteins. Now, RNA controls the assembly of the amino acids that form the proteins. So each type of RNA molecule specializes in a different aspect of this job. Okay, so there are, again, 3 main types of RNA. You see them here. They are messenger RNA, also known as mRNA. There's ribosomal RNA, known as rRNA, and then transfer RNA, which is tRNA. Now, RNA is very important. As DNA is the inherited genetic material, RNA is the genetic material that carries out the instructions that are encoded in the DNA. So without RNA, the instructions in the DNA couldn't be used by the cells. It would be worthless. So RNA is incredibly important. So let's talk about each of the different types of RNA. We'll start with messenger RNA or rRNA. And many DNA genes contain the instructions for assembling amino acids into proteins, and the RNA molecules that form as copies of these instructions are called messenger RNA. So RNA is kind of the generic term. I like to think of RNA as being um, the strand of RNA that's inside of the nucleus. Once the RNA leaves the nucleus, then it's considered mRNA because now it's carrying a message. That's why it's called a messenger. Messengers carry messages. So this messenger RNA is carrying a message to ribosomes that are going to use their ribosomal RNA to know how to match up tRNAs with the bases that are in the mRNA strand. We'll get into how that all works. Don't let it confuse you too much. Just know that the RNA molecules, once they leave the nucleus, now they are considered messenger RNA or mRNA, and they are really providing the instructions. So for a polypeptide synthesis process, building proteins, the messenger RNA moves from the nucleus to the cytoplasm where it encounters a ribosome. Now, the second type of RNA is called ribosomal RNA or rRNA. Now, proteins that are assembled on ribosomes, those ribosomes are basically small little organelles, and they're composed of two subunits. So these subunits are made up of several ribosomal RNA molecules, and they can have as many as 80 different proteins that make them up. Now, transfer RNA, when the protein is being built, this RNA, the transfer RNA, um, transfers each amino acid to the ribosome as it is specified by the coded messages in the mRNA. These molecules we call transfer or tRNA. So, how does the cell make mRNA or RNA? Well, most of the work of making RNA takes place during the process of transcription. So, in prokaryotes, RNA synthesis and protein synthesis both take place in the cytoplasm. But in eukaryotes, which have a distinct membrane bound nucleus, the RNA is produced in that nucleus, and we see that here in the illustration. So you can see that we are currently inside of the nucleus. See, the nucleus is listed here. You can see a portion of the nuclear membrane right here. Okay, so that shows us that we're inside of the nucleus. Okay, and once that RNA is made, it's going to leave the nucleus and enter into the cytoplasm of the cell. Now, this process of building that RNA molecule is called transcription. So there's two words that deal with creating complementary strands or utilizing the information in the DNA, I guess is a better way to say that. One is transcription, and that is the process of the DNA, one of the DNA strands, a section of it being copied to form an RNA strand. Translation, we're going to get into later. Translation is when the actual amino acids are brought in and linked together into the polypeptide chain. That's called translation, and that happens in the cytoplasm. But in the nucleus, we call this transcription. It's important to know the difference between those two terms. So, transcription requires an enzyme, and it is known as RNA polymerase. You see it circled here now. It's the golden globe there behind the DNA and the RNA strand that's being formed. And the RNA polymerase binds to the DNA during transcription and separates those two DNA strands. And you can see that those two strands are separated here. So we have replication forks on this side and a replication fork over on this side as well. So this is sort of like unzipping the strands of DNA, right? RNA polymerase then uses one strand of DNA in order to use that as a template from which it will assemble the nucleotides into a complementary strand of RNA, which will be the mRNA. And we can see that this RNA strand is being formed here. So the ability to copy a single DNA sequence into RNA is what makes it possible for a single gene to produce hundreds or even thousands of RNA molecules. So here's a question for you. How does RNA polymerase know where to start and stop making a strand of RNA? Well, if you're thinking that RNA polymerase doesn't bind to DNA just anywhere, you're right. The enzyme only binds to what we call promoters. The promoters have a specific base sequence, and that base sequence tells the enzyme where to start transcribing DNA. So keep in mind as we continue along here that transcription is the first stage of RNA synthesis. So what happens to the newly made mRNA before it leaves the nucleus? Well, it's called RNA editing. So the new mRNA molecules are like a writer's first draft. So sometimes they require a little bit of editing before they're ready to be read. So the pieces of pre-mRNA, this is what I like to call RNA, but we got to give them a bunch of fancy names. But the pieces of pre-mRNA molecules that are cut out or edited out and discarded are called introns. Okay, so that's the editing piece. They're cutting out pieces of unnecessary code, and these pieces are called introns. So here are the introns here. They're basically removed and tossed. The remaining pieces that are left are called exons. Okay, here are the exons, those smaller, darker blue sections. Okay, those exons are basically spliced together, and they end up forming the mRNA. So it's safe to say that this pre-mRNA is actually just what we can call RNA. And once it gets edited up, it's now mRNA and ready to take off out of the nucleus and go build proteins. All right, so after the exons get split together, there is one more step that has to occur, and that is the RNA caps and tails have to be added to this final form of the mRNA molecule. So you see a cap here and a tail there. So what is the purpose of making a large RNA molecule and then throwing out parts of the molecule that aren't needed? Well, that's a good question. Biologists still don't quite have a complete answer. Some pre-mRNA molecules might be cut and spliced in different ways in different tissues. So a single gene can actually produce several different mRNA molecules. That could be one explanation. Another one might be that introns and exons both play a role in evolution, making it possible for very small changes in the DNA sequences to have dramatic effects on how genes affect cellular function. Now, they're not sure that that's true. That's just something that they think might be an explanation, but they're still working on trying to figure that all out. And that is how RNA is made.